Popular culture loves to compare the human brain to a computer. We picture a rigid, highly organized circuit board inside our skulls, processing information through a sequence of digital switches flipping definitively on and off. But this analogy breaks down when we look closely at cellular biology. A neuron does not operate as a binary switch. Instead, neural communication relies entirely on a fluid electrochemical process. The interior of a cell maintains a different concentration of charged ions compared to the space outside of it. This animated graph plots a neuron's cellular voltage. Because of unequal ion distribution, a resting neuron maintains a steady difference near -70 millivolts. Yet this resting state is never truly still. Constant background inputs cause its voltage to continuously jitter and undulate. To understand how the brain actually processes information, we have to let go of the digital computer metaphor. The mind operates in a chaotic, constantly shifting electrochemical environment. The rules of this environment are dictated at the synapse, the specialized junctions where neurons connect and influence each other's electrical states. When an upstream neuron becomes active, it releases chemicals that trigger changes in its neighbor. Excitatory inputs are signals that push the receiving neuron's internal voltage higher, driving it closer to its own point of firing. Inhibitory inputs do the exact opposite. They push the internal voltage lower, driving the neuron further away from firing. This diagram shows a single central neuron receiving competing incoming signals. A neuron's state at any given millisecond is the continuous mathematical integration of these opposing forces. Its voltage rises and falls based strictly on the exact ratio of the inputs it receives. When that integrated voltage finally hits a critical threshold, it triggers an action potential. This is a rapid, self-propagating electrical spike, the moment a neuron finally fires. But watch the threshold line itself. While the action potential is an all-or-none event, the threshold required to trigger it is constantly moving, adjusting based on the neuron's recent activity and internal chemistry. Because this threshold is a shifting target, a neuron acts as a dynamic integration engine. It constantly weighs competing electrochemical votes rather than waiting like a rigid mechanical trigger to be pulled. Because of this dynamic environment, some of the most critical work a neuron does happens quietly, long before it ever generates a spike. This is subthreshold neural activity. It consists of continuous, rolling waves of voltage changes that actively shape a neuron's probability of firing in the future. These columns represent the internal voltage of two distinct neurons. The one hovering right beneath the threshold line requires only a tiny fraction of incoming stimulation to fire, compared to the one resting far below it. If we expand this concept out across a broader network, we see a vast topographical surface of peaks and valleys. Neurons ride these rising and falling voltage waves, and their position in this rolling landscape dictates exactly which cells are primed to act next. Neural computation relies heavily on this unseen tension. The cells that are primed and ready to fire exert as much influence over our cognitive processing as the cells that are actively firing. Individual neurons do not produce perception or behavior on their own. These functions only emerge when massive populations of cells coordinate their activity. Across these immense networks, millions of cells exist in varying states of subthreshold activation at the exact same time. Because the brain has finite metabolic resources, these different neural populations must fight for expression. When a specific network becomes active, it physically amplifies its own related activity while actively suppressing the firing of competing networks. The macroscopic view of the brain is a continuously evolving arena. Representations of memories, incoming sensory inputs, and motor programs battle against one another to dominate your conscious attention. Modern neuroscientists are actively working to map this landscape and decode the exact mathematical rules governing this competition. One proposed structural framework is tokenization theory. It investigates whether these continuous competitive dynamics might organize into discrete, recognizable units to drive broader cognitive functions. However, while the biological mechanisms of excitation and inhibition are well documented, they provide only a foundation. They do not inherently prove the specific macroscopic claims of tokenization theory, which remains a subject of ongoing exploration. What is strictly proven by the biology is that your mind does not run on rigid digital switches. It is built entirely on a restless, continuous electrochemical competition, where even the quietest fluctuations dictate how you experience reality.